Hi there. This video is going to be about the cells that make up the middle layer of the heart, our cardiac myocytes, the cardiac muscle that does the work of the heart. Now, cardiac myocytes, and myocyte just means muscle cell. Cardiac myocytes are short and branched. If you recall, in skeletal muscle, we had really long muscle fibers that went all the way from one end of the muscle to the next. Here, we're not trying to get strength over a long distance. We're trying to get strength and flexibility in a short, round area. So the cells are shaped differently. They're short and they're branched. So the different cells can connect to multiple other cells. It's a way to form this dense network, which we'll see actually helps them to communicate and contract in a unified manner. So, cardiac myocytes, wherever they connect to another cell, they form what are called intercalated discs. They're these zigzag shapes. You can see the dark lines here marked by the arrows, sort of where each cell has interdigitating folds that come together so that they have the maximum amount of surface area. One other thing I want you to look at here, two other things really. Remember that just like in skeletal muscle, we have the thick filaments and the Z discs and the thin filaments. We have the same mechanism here for the muscle to contract. And these blue structures are mitochondria, and they're producing ATP to power the heart. I think, when you look at how many mitochondria there are making ATP for these heart muscles, you get a sense of just how much energy it takes to keep our heart going. Okay, let's take a closer look at these intercalated discs. So here the artist has taken away the adjoining cell so that we can see the folds of the membrane where the two cells would connect. And there are these pores or channels here that actually go through the membrane of both cells, right? So here's one cell in the membrane, here's the fluid in between, here's the membrane of the invisible cell, and you can see that there's a pore or a channel that goes directly from the inside of one cell to the inside of the next cell. Here's another way of looking at it. Here are the two cells, and there's a channel that connects the inside of one to the inside of the other. This is different from the channels that we've seen so far that are connecting the inside of the cell to the outside in order to do an exchange with the outside environment, move ions or fuel molecules. This is directly cell to cell, and this is super important in the heart. So, just like you saw in skeletal muscle, heart muscle, in order to contract, has to start with an action potential. Now, in skeletal muscle, we had a neuron coming from the nervous system connecting to the muscle fiber itself, and then it would start the action potential by sending a neurotransmitter across to the synaptic cleft. And you kind of remember these steps, right? It was a lot. So, in the end, and this is true for both cells, this action potential causes the release of calcium inside the cell, and that calcium is what acts as the trigger for muscle contraction. Now, here's where calcium is doing its job. You may recall that the thin filament, the rope that the big, muscly, thick filaments are pulling on to shorten the sarcomere, the thin filament is actin, and it's covered with something called tropomyosin. And tropomyosin is covering the parts of actin that the myosin head groups can connect to pull. So it's like covering the handles so that there's nothing to grab onto. But when calcium shows up in the cytoplasm, it binds to this molecule called troponin, And troponin signals tropomyosin, get out of the way, uncover the handles. And it does. So once calcium binds, troponin tells tropomyosin to get out of the way. Now we can see the active sites, and now our little muscles, muscle fibers. I guess they're not muscle fibers, but they're like the muscle of our muscle. These myosin head groups bind to the active sites and can start pulling, right? So we have the same thing in cardiac muscle. Here, just to remind you, is where the thick filaments are. The thin filaments are these orange ones. So the thick filaments grab onto these ropes and pull. The Z discs get closer together and the muscle shortens. And then we have to go through this process of getting it to let go. And to think that the heart's doing this at least once every second. Okay. So the same stuff about calcium and troponin and tropomyosin and the active groups in the thick filament, all of that is still true in cardiac muscle. The main difference between these two types of muscle is how the cardiac myocytes are stimulated to have action potentials. So let's talk about that. Action potentials for the heart are initiated from within the heart, not by the nervous system. So we're going to talk about this soon. This is called the SA node, or the sinoatrial node, and is known as the pacemaker of the heart. So instead of having neurons coming from the nervous system to make something work, it's all handled internally. The other thing is that action potentials, which is really just calcium and sodium showing up inside the cell, are passed directly from one cell to the next through gap junctions. Instead of having a neuron release a neurotransmitter and get to a ligand-gated channel that starts a local potential, then an action potential. We don't have time for that in the heart. In the heart, when this cell is triggered to have an action potential, so calcium and sodium come in, that's calcium and sodium are going to, at the same time that they're causing the changes here, move to the next cell and start an action potential there. So it's sort of like a traveling action potential, local potential, action potential, but it just happens so fast because they're connected. It's like one giant cell in a way. Okay, so let's talk a little bit about some of these specialized parts of the cell. So what I want to look at next are ventricular myocytes. All right, so that means we're looking at the muscle cells that make up the ventricles. And they have a special action potential that allows them to hold their contraction for longer than what a skeletal muscle fiber does. This allows the ventricles to be able to eject more blood with each contraction. So if you imagine that you're holding a tube of toothpaste and you squeeze it quickly and then let go, like a quick twitch, then you're going to get a little squirt of toothpaste that comes out. But if you take that tube of toothpaste and you squeeze just as hard, but you hold it, the toothpaste is going to keep coming out. You're going to make a bigger mess. And that's the idea of having the ventricles hold their contraction for longer. So let's look at how that works. Okay, so the way that the action potential begins is that it's coming from through a gap junction from a neighboring cell. That positive charge coming in from the neighboring cell causes these two channels to open. So this is a sodium channel and this is a calcium channel. Now they both open or trigger to open more or less at the same time, but sodium channels are really quick to open and close, right? So it's a very sharp action. Calcium channels open and close slowly. In these cells anyway. So picture like, a young person sitting next to a really old person at a sports stadium and the ball is coming and they have a chance to catch it. The young person can, stand up quickly and do it, and the old person is still trying to get out of the seat. And same thing for sitting back down. Okay, so what that means, if we look at this green line, is that as soon as the action potential arrives, the calcium and sodium come from the gap junction, and these two channels open, we get a super sharp, super fast increase in the charge in the cell. It becomes way positive. Okay. And then when we reach this point, what's happening is that the sodium channel is closing. So the calcium channel has just gotten up. Now the sodium channel is already closing and sitting back down. And so we do have the charge becoming more negative again for that reason. But also it doesn't go right back down because the calcium channels are still open. They haven't gotten back to their closed position. So if it were sodium by itself, it would be a sharp spike like this and back down. And if it were calcium by itself, it would be a sort of a smooth wave. And when you add the two of them together, this is what you get. The important point is, and also, I think I forgot to put my repolarization. We'll get there. But the important thing is, if we look at the red line, which is where the muscle's actually contracting, it's contracting for a longer time. It's not just having a quick twitch, if you remember what that looked like for our skeletal muscle fibers. So this is how they do it so that all of the cells together can hold that contraction for longer. So the longer that we have calcium in the cytoplasm, the longer the contraction is going to last. So having these slow calcium channels is what gets us there. Okay, to repolarize so that we can do it again, we use potassium channels, just like we do in every other electrically excitable cell. We let potassium leave the cell because remember, it's the only ion that's most concentrated in the cell. So it wants to leave. As it leaves, we lose positive charges and the membrane potential becomes more negative again or returns to its resting membrane potential. Okay, and so this action potential that we just created, just like it has everywhere else in the heart, is traveling from cell to cell through gap junctions. So sodium and calcium come in here. We can say that this is the SA node having an action potential. Sodium and calcium, in addition to causing the contraction here, also diffuse through the gap junction to their neighboring cell. That causes these two channels to open because they're voltage gated. They let in more sodium and calcium. That moves to the next cell and so on. Okay, so there are actually several heart structures that are capable of initiating these heartbeats, where the signals are passing through the gap junctions from cell to cell. And that's what we're going to talk about next. How do they do it? These three structures are called the SA node, the sinoatrial node, because it is in the atrium, the sinus of the atrium. Sinus means sort of opening or whole cavity. And the AV node, which is right here, that stands for atrioventricular because it's located between the atrium and the ventricles, more or less. And the ventricular myocytes themselves. All of these are capable of initiating a heartbeat, meaning that if the SA node fails, then the AV node will take over and continue to make the heart contract. If this fails, then the ventricles themselves, the individual cells, can do the same thing. Now, the reason that the SA node is considered the pacemaker is that it actually has the fastest rhythm, which means that before the AV node or the ventricular myocytes can get to the point where they send out a signal that it's time to contract, the SA node has already done it. It's moved on to its next stimulus. So because it's fastest, it gets there first every time in a healthy heart. All right, so before we talk about how that whole system works, let's take a look at the cells of the SA node and how they do their thing, keeping in mind that the same thing's happening here, just much more slowly. All right. Now, The resting membrane potential, which you learned about in the previous semester of this course, is the membrane potential that is developed by the sodium pump or the sodium potassium ATPase and potassium channels, and it's essential in electrically excitable cells. What those two membrane proteins accomplish is that they build a high concentration of potassium inside the cell and a low sodium concentration. So they're getting rid of sodium and they're bringing in potassium to create this gradient. Also, they make the inside of the cell more negative because every time they release positive ions, the inside of the cell has a more negative balance or has fewer of the positive ions. Okay, so this is the background that we've got for the cell to work. Now, the cells of the SA node have these sodium channels that are leaky. That means they're never all the way open and they're never all the way closed. All right, so we've got this negative cell here, and the sodium is just sort of dripping into the cell. If we put an electrode in the cell and we measure the charge inside the cell compared to the outside, then we would see that it's slowly becoming more positive, right? As this sodium, this positive sodium, is leaking into the cell. Now, when it reaches a certain charge, which we call the threshold, when it reaches the threshold, which is the same thing as the voltage that will open this channel, then the calcium channels open. Let me move me around again. Right? So the sodium was leaking in here. When we reached this charge, the calcium channels opened. Calcium comes rushing in, and it's got two positive charges for each ion. And so it gets positive very quickly. And then when the membrane potential reaches its peak, then the potassium channels open and the membrane potential returns back to the resting membrane potential. And this little blip is what we call the action potential. But what makes it happen over and over on its own is this sodium that constantly leaks in and gets us to the point where the calcium channel reacts. If we were in skeletal muscle, we'd be waiting for a signal from a motor neuron. Right? And once again, remember that calcium is traveling through these gap junctions to the neighboring cells, which is causing them to repeat or amplify the action potential and pass it on to the next one. And diffusion is super fast. All right, so the whole process begins again because we have the leaky sodium channels. They're actually leaking the whole time. They don't stop just because we're having an action potential. It's just that the charge changes rapidly with these other channels suddenly being active. All right.